is the phosphorus balance in muscle tissue and muscle function in dairy cows, undergoing dietary phosphorus deprivation during the transition period. Thank you, doctor.
Okay, thank you for the introduction. We already heard that hypophosphatemia is a common metabolic disturbances we see in dairy cows, particularly around calving. The problem is we don't really understand what the clinical relevance of this is. There is some empirical evidence suggesting that there is indeed an association between hypophosphatemia and recumbency. However, if we try to understand how this works and what the underlying mechanism for this would be, we didn't identify one yet. And the other important point is that thus far, there was no way to experimentally induce hypophosphatemic recumbency in cattle yet. So our hypothesis for the study I'm presenting today was that we would be able to induce some sort of muscle disturbances by inducing hypophosphatemia. So the objective was to experimentally induce hypophosphatemia by feeding a phosphorus-deficient ration during the periparturient period, and to see, A, how this affects the phosphorus content in muscle tissue, and B, how this affects muscle function on a clinical and subclinical level. So what did we have? We used 36 late pregnant dairy cows for this study. They were approximately 6 weeks before calving when enrolled, and we assigned them to one of two groups. We had the control group, animals that received a ration with adequate phosphorus supply during the entire study, and we had a low phosphorus group that was fed a phosphorus deficient ration from 4 weeks before to 4 weeks after calving. So essentially we had 3 experimental phases in our experiment. We initially had an acclimation period that started 6 weeks before calving, that went to 4 weeks before calving, identical for both groups, where we fed a standard ration with adequate phosphorus supply. Then we had the depletion period that went from 4 weeks before calving to 4 weeks after calving, where the low phosphorus cows were fed a phosphorus deficient diet. And then for a subset of cows, so pretty much half of the cows of each group, we followed up with a repletion period here, during which we supplemented phosphorus in the low phosphorus group to see if we could reverse some of the effects we may have caused by decreasing phosphorus in the diet by supplementing it in the end of the study. Here I'm giving you the phosphorus contents we used in our experiment. So you see for the group C, a dry cow phosphorus content was around 0.28%, and in lactation 0.44. So that is essentially according to official recommendations. When we look at the low phosphorus during the acclimation, we had the same as in the control group. Then dry cow depletion 0.15, so that is as low as we could get. And then after calving, 0.20% phosphorus in dry matter. And then during the repletion, we switched the low phosphorus cows again to the ration of the control groups. What did we look for? The animal behavior. Animal behavior, demeanor. We tried to see signs of muscular weakness. This was done daily. We took blood samples. You can see here, I wrote jugular vein. I do not really agree that you need to do the coccygeal vein, saying the important thing is you do it consistently, so you can say you can follow up the changes here. Yeah? This was done once a week. And then as far as muscle tissue was concerned, we took muscle biopsies from the biceps femoris muscle of the hind limb, and we did electromyographies on the biceps femoris as well and the intercostal muscles. These muscle analyses were done at 5 specific time points, T1 to T5. These were T1 at the end of the acclimation period, that was our baseline. Then after 2 weeks of phosphorus depletion, T3 in the first week or during the first week of lactation, T4 at the end of the depletion period, and T5 for those cows that underwent the repletion period at the end of the 2-week repletion period. The parameters we analyzed, I'm going to present in plasma, the plasma phosphate. And then for muscle tissue, we specifically I'm going to present the results for muscle tissue phosphorus in wet weight, but we're also going to look at another quantitatively important mineral, which is potassium in muscle tissue, and you will see why in a minute. And we also looked for tissue dry matter. As far as electromyography was concerned, we had myologists from equine on board, so we did the electromyography on the biceps femoris and intercostal muscles, and here we specifically focused on so-called pathologic spontaneous activity and on motor unit action potential analysis. So I'm getting to my first results slide. This is just to show you that our intention to induce hypophosphatemia by feeding a phosphorus-deficient diet actually worked. So you can see here the open circles. This is the control group, remained within the reference range during the whole study, whereas the low phosphorus group, these are the closed circles, there was a continuous decline, reaching a nadir at the time of calving with mean values below 0.4 millimoles and remaining at that level until the end of the depletion period. After that, the values went right up. So the diet, that worked really well. Now, clinically, what did we see? Up to the point of parturition, really no difference whatsoever between the two groups. Then at the time of calving, most important question, did we have recumbent cows? Yes, we did. Unfortunately, they were all in the control group. So there's obviously an association between phosphorus and calcium, but this is a topic you will hear about in the next presentation. As far as the low phosphorus group is concerned, there was no difference or no signs of muscular weakness in this group. What we did see, though, were a number of cows that developed post-parturient hemoglobinuria. This is another condition that has been associated with hypophosphatemia in fresh cows. And this was also the reason why we had to remove 3 of the 18 cows from the study in order to be able to treat them properly. Now I come to the results of the muscle biochemical analysis. The first thing I'm showing you here is the phosphorus content in muscle tissue wet weight. So you see the different time points and you see the boxes. Purple is low phosphorus, green is control. And what you can appreciate here is that the phosphorus content in muscle tissue remained constant during the entire depletion period. In contrast, you see that at the end of the depletion period in the control group, you have a decline, but you have a similar decline, somewhat time delayed in the low phosphorus group at the end of the repletion period. And the interesting thing is, if you look at potassium in muscle tissue, it looks exactly identical. So there is apparently something happening, but it's affecting both groups, and it's not just affecting phosphorus, it's also affecting another intracellular electrolyte, which is potassium. And you get a grasp about what the underlying mechanism might be if you look at the development of tissue dry matter. There is a decrease of muscle tissue dry matter at the end of the study in the range of 10 to 15%. And this is probably the mechanism, which means decreased tissue dry matter means increased tissue water, which means increased volume of distribution for intracellular electrolytes in this dilution. Let's move on to the electromyographic results. So here, the first thing is pathologic spontaneous activity. This is a bit tricky. Essentially what you do is you stick a needle in the muscle and you see if you can induce spontaneous discharge. This is PSA, pathologic spontaneous activity, and this is something that is suggestive of what we call neuromyopathy. Neuromyopathies may occur because of membrane instabilities that can occur either at the muscular or at the nerve level. So what you see here is we determined the occurrence of pathologic spontaneous activity. You can grade this as either mild, as moderate, or pronounced, depending on the amount of discharges you get. And what we're presenting here is the percentage of cows that showed either mild, moderate, or pronounced PSA at the different sampling times for control and the low phosphorus group in here for the biceps femoris muscle. So obviously what becomes apparent is that we have a change of time, so increased mild PSA in both groups. We attributed this to the fact that we had to keep the cows in tie stalls, so they had limited possibility to exercise the muscle. Yeah? But what also becomes apparent that moderate and pronounced PSA is only seen in the low phosphorus group at the end of the study. If we look at what happens in the intercostal muscles, we find exactly the same pattern, just more pronounced. We have some mild PSA from the beginning on. This increases over time, but again, predominantly in the low phosphorus group, we find the animals with moderate and pronounced PSA toward the end of the study. So from this, we conclude that evidently there is a time effect in both groups, but we see the severe case is predominantly in the low phosphorus group. We see that apparently there is a difference between muscles, so it seems that intercostal muscles are more susceptible to these effects. And the other important thing is it appears that these effects do not appear to be corrected by phosphorus supplementation during 2 weeks. So apparently these effects are sustained for longer despite of correction of hypophosphatemia. The other point is the electromyographic results of the motor unit action potential analysis. This is essentially the action potential you measure that is associated with muscle contraction. And what we do here, we have a number of parameters that are included in this analysis. And crudely speaking, if you have any of these parameters that decreases in amplitude, this is consistent with decreased muscle function or decreased muscle weakness. So what did we get here? If we look for the biceps femoris muscle, we had only one of these parameters that decreased. That was essentially the size index, and it decreased at T4, which means at the end of the depletion period and returned to normal after phosphorus repletion. So there was a mild effect apparently affecting muscle function. This could be either that fewer fibers are contracting or that the fibers per se were weaker. We can't comment on that. If we look at the same in the intercostal muscles, we had several parameters that changed there. So not just the size index, but we had several parameters, and they already changed at T3, just after calving. So we had an effect there. The effect occurred earlier, was more sustained, but again, was at least partially reversed by supplementing phosphorus. So what do we get from all this? So clearly what we can say, we're really good in inducing hypophosphatemia. So decreasing the dietary phosphorus very effectively causes hypophosphatemia. We had several cows that had phosphorus concentration below detection limit for weeks, and you wouldn't see anything on them. But these changes in plasma phosphorus were not sufficient to induce changes in muscle tissue phosphorus, which means that the muscular tissue per se is apparently very effective in maintaining the intracellular homeostasis. But we clearly have to admit, even though we did not see any clinical signs, at a subclinical level, undoubtedly, there were effects on muscle functions. We had two different types we found. One thing is neuromyopathies. That could be associated with membrane instabilities. Keep in mind that membranes, cell membranes, are composed of phospholipids, and that these neuromyopathies are not easily reversed. So they may be sustained for longer. And the other thing is muscle fiber function. This could be associated with muscle weakness. And these effects could be reversed relatively easily. And apparently there are differences between muscles. So we saw the most prominent effect on intercostal muscles. That might be dependent on the type of fibers you have in the different muscles. But we saw very, very mild effects on the hind limb muscles, where we would expect to find most issues. So clearly, phosphorus deprivation was associated with subclinical signs, but was not associated with any clinical signs. So I'm personally struggling to use this as an explanation for the clinical signs that have been proposed based on the clinical observation. But I'm certainly convinced that the effects we saw could be causing disturbances of health and well-being in the cows. So certainly something to keep in mind in future studies. Thank you very much.